like to bring everybody's attention for this afternoon's exciting Wednesday afternoon lecture that today is going to be given by Sandy Schmidt. So Sandy Schmidt is the chair of the Department of Cell Biology at Scripps and has been so since 2000. I've had a really wonderful opportunity of knowing Sandy for many years. In fact, since she was a graduate student with Jim Rothman at Stanford. And I can say that Sandy has, through every step of her career, really demonstrated an excellence in science with true leadership and mentoring skills. Sandy is most widely recognized as a really leader in the field of endocytosis. She was really the first to introduce an in vitro assay for analyzing endocytic dynamics, and that quickly led her to really to dynamin as a central regulatory GTPase for this whole process. And by integrating in vitro approaches with her structure function analysis of dynamin and its role in endocytosis, she really opened up the field to many different aspects of how this very important endocytic portal into the cell is regulated, including the role of small GTPases like RAC and Rho, as well as actin. Now, more recently, and this is what we're going to hear about today, Sandy has been integrating various new biophysical approaches for dissecting this really important process within cells. In particular, she's used total internal reflection microscopy to look at the actual budding of these endocytic vesicles, as well as planar supported bilayers to generate an artificial system where she can really dissect how all of this machinery is operating, in particular the role of cargo in regulating dynamin activity. Now, as a result of really this spectacular body of work that's characterized Sandy's work as a scientist, she's received many honors. She was elected, she was given the William Rose Award for the ASBMB. She's currently a fellow of the American Association of Advancement of Science. And most recently, she's been elected as the president of the American Society for Cell Biology. Now, coupled with all of this scientific and leadership role, Sandy has played a huge role in the publication arena in cell biology. She spearheaded the introduction of the new journal, or the journal Traffic, which is not new anymore, but she started it from scratch, and it was based on her superb running of that journal that she was then selected as editor-in-chief for the molecular biology of the cell, which she's only recently given up in lieu of her presidency to the American Society for Cell Biology. So I should mention that Sandy received her BS at the University of British Columbia. I mentioned she got her PhD at Stanford, and then she postdoced at Yale with Ira Mellman before moving to Scripps in 1988, where she has been there since. Currently, she's serving the role of chairman of the cell biology department there. So with that, I'd like to bring Sandy up to the podium, and she's going to talk to us about protecting our borders, regulated entry into the cell.
Thanks, Jen. Thank you very much. It's a pleasure to be here and to see many old and new friends. I have not visited NIH, the research laboratories before, so long overdue, and I hope I get to come back again soon. Maybe give a group meeting or something. The plasma membrane is the barrier that protects the delicate intracellular environment from the harsh extracellular milieu. It's also a sophisticated communication device, which is important to keep in mind. Signaling receptors on the cell surface are responsible for all of the communication of cells to cells and cells in development and motility, chemotaxis, physiological changes that happen. These are all mediated through communications at the level of the plasma membrane. It's also a dynamic port of entry. There are multiple endocytic pathways that govern nutrient uptake, surveying of the extracellular milieu in the immune system, turnover of plasma membrane transporters in response to the physiological changes when you eat or when you have a high glucose in your bloodstream. All of these processes are being controlled by a myriad of endocytic pathways, each mechanistically distinct, and each one tightly coupled to and regulated by the specific cargo molecule that is being taken up by these processes. Today's talk is going to focus on clathrin-mediated endocytosis, which is the major endocytic pathway in the cell. It's certainly the best understood endocytic pathway in the cell, and it is involved in the specific internalization of receptor and concentrative internalization of receptor ligand complexes. Clathrin-mediated endocytosis occurs at specialized regions on the cell surface. In the background of this slide are electron micrographs taken by John Heuser that show the polygonal lattice of the clathrin coat that forms these clathrin-coated pits. The process involves the assembly of adapter molecules. Juan Bonifacino has studied them to great extent that recognize small, short sorting motifs in the cytoplasmic tails of receptors that are going to be concentrated in coated pits. These adapter proteins also trigger the assembly of clathrin, which is a self-assembling scaffold that forms into a polygonal lattice, helping to generate curvature in the growing vesicle. Here. The protein dynamin that we're going to talk about is targeted very early on during vesicle formation and eventually assembles at the neck. We'll talk a lot about that, leading to finally a fission reaction and the production of an endocytic clathrin-coated vesicle that carries a concentrated load of receptor ligand complexes, cargo, into the cell. This spontaneous assembly of clathrin and adapters needs activity and energy to disassemble, and so the encoding ATPase that I discovered as a graduate student in Jim Rothman's lab, it's since been studied in great mechanistic detail by Lois Green and Evan Eisenberg here at NIH, mediates an uncoating reaction to recycle coat proteins for multiple rounds of endocytosis. In addition to these major constituents of the coated vesicle machinery, there's a plethora of other endocytic accessory factors, most of which have been identified through protein-protein interactions with adapters, clathrin, or dynamin, which serves as hubs in this systems biology type look at endocytosis by Harvey McMahon and his colleagues. Specific lipids are also required, and of course, the cargo is an important regulator. The question that one asks is, why is endocytosis so complex? Why all this myriad of accessory proteins when you compare it to vesicle formation from intracellular organelles that have been well studied by Jim Rothman, Randy Sheckman, Bill Balch, and others, where they have been able to reconstitute in vitro systems COP2 or COP1 vesicle formation using just the coat proteins, Sec23-24, sorry, I should have, Sec13-31, or the coatomer complex, and the small GTPases, SAR or ARF. So why the complexity of endocytosis at the plasma membrane? Now, you might argue it's because the plasma membrane is more rigid, it has higher cholesterol composition, it's got the underlying cytoskeleton, it's got the extracellular matrix, but I would say that there are multiple endocytic pathways that don't require this plethora of proteins. So rather, I would argue that the complexity reflects the need for strict regulation and these higher order communication functions of the plasma membrane. So key regulators of vesicle formation at each of these stages are members of the GTPase superfamily. The RAS-related GTPases, SAR and ARF, regulate vesicle formation at the ER and the Golgi, respectively. And the main gist of this talk is to tell you about dynamin, which regulates vesicle formation at the plasma membrane. So dynamin is both the brains and the bronze of clathrin-mediated endocytosis, and that's one of the main take-home messages of this talk. It was first discovered, its role in endocytosis was first discovered in 1973, actually at my alma mater at the University of British Columbia by David Suzuki, who was conducting temperature-sensitive screens for chemically induced mutagenesis in flies. And he set up a screen for paralysis and isolated the shibiri flies. This is Japanese for paralyzed, providing the first evidence for involvement of dynamin in endocytosis. Bing Zhang at UT Austin made this movie for me. These are mutant flies and wild-type flies being subjected to ever-increasing temperature. And you can see as the temperature gets above 30 degrees, the mutant flies drop like flies, whereas the wild type are unaffected. This is a rapidly reversible phenotype. Jane Koenig and Kazuo Ikeda at the City of Hope in California studied these flies by looking first, not surprisingly, since they had this paralyzed phenotype, at the neuromuscular junctions. And at the neuromuscular junctions of these flies, you can see at the non-permissive temperature the typical morphology of a synapse, neuromuscular synapse, chock full of synaptic vesicles and docked here at the muscle. Whereas just 5 minutes after shift to the non-permissive temperature, you can see the synaptic vesicles have been completely depleted, and instead you see this accumulation of endocytic profiles on the presynaptic membrane. At higher magnification, Kazuo Ikeda noted that many of these endocytic structures have an electron-dense band at their neck. He called them collared pits. Some years later, shibiri was cloned by Richard Valley and Alex Vanderbleek at the same time and discovered to be the homologue of a mammalian protein that Richard Valley had discovered and called dynamin. Dynamin is atypical GTPase. In addition to the standard RAS-related G domain, it has a middle domain and Z domain that are involved in self-assembly, a pH domain that interacts and targets to PI45P2 containing membranes, and a C-terminal proline-arginine-rich domain that interacts with several of these endocytic accessory proteins that are SH3 domain-containing molecules. Dynamin is a tetramer in solution, so if you think of RAS as small GTPases and trimeric G-alphas as large GTPases, you can think of this as the Arnold Schwarzenegger of GTPase, or not to mix metaphors, the Incredible Hulk of GTPases. It has a low affinity for GTP in the 10 micromolar range. The consequence of this is, unlike RAS-related GTPases and trimeric GTPases, it does not need a GEF to activate it. It does not need a guanine exchange factor to yank off bound GDP that is so tightly associated so that it can be loaded with GTP. Rather, in the cell at steady state, it will always be in the GTP-bound form. It also has a robust basal rate of GTP hydrolysis of about 1 per minute. Dynamin self-assembles onto rings and spirals in low salt solution or in the presence of GDP aluminum fluoride. This is a discovery that Jenny Henshaw made when she was in my lab at Scripps. And onto liposomes, either acidic or PIP2 containing liposomes, to form these long dynamin spirals, very reminiscent of the shibiri collars seen in the Drosophila mutants. This self-assembly stimulates dynamin GTPase activity about 100-fold, and mutations in the GED, the GTPase effector domain, impair self-assembly and decrease influence dynamin's GTPase activity. So based on Jenny's discovery that dynamin self-assembles into these collar structures on its own, and previous work in my lab using the in vitro assay that Jennifer had referred to, where we had shown that you accumulate, new clicker changer, that you accumulate constricted coated pits in the presence of non-hydrolyzable analogs of GTP. Jenny and I proposed that dynamin functioned as a mechanochemical enzyme that upon addition in the presence of GTP, dynamin would self-assemble into this collar-like structure at the neck of deeply invaginated coated pits. And if dynamin underwent a coordinated GTP hydrolysis, these assembled subunits would undergo concerted conformational change that together could generate the force necessary for scission. However, to test this model, we made mutations that were specifically defective in this self-assembly reaction and assembly-stimulated GTPase activities. This work by Sonia Sever in my lab. And unfortunately, when we put these mutants in vivo to look at their phenotype, we got exactly the opposite phenotype that our model would have predicted. That is, overexpression of these mutants that selectively impair self-assembly and therefore assembly-stimulated GTPase activity actually accelerated the rate of transfer and clathrin-mediated endocytosis in these cells. So that led Sonia Sever and I to propose another model for dynamin, in which we proposed that dynamin was actually like every other GTPase in the world, not a mechanochemical enzyme, but indeed a regulatory enzyme. In this model, we suggested that dynamin must, in its GTP-bound form, interact with effector molecules that control the rate-limiting steps of vesicle formation, so that if we make mutations that are impaired in self-assembly and therefore in the gap activity, we would prolong the interaction of dynamin with these effector molecules, accelerate the rate-limiting steps of vesicle formation, and that self-assembly and GTP hydrolysis, which activated the gap, in a sense, would, like other GTPases, serve to negatively regulate activity and turn off the molecule for recycling. So these are apparently conflicting models, embarrassingly both suggested by me, but thankfully both published in Nature. So this gave us lots of chance for reviews. But what I'd like to suggest is that, like this optical illusion of an older and a younger woman, that these two models for dynamin are not mutually exclusive. So we think now that dynamin plays a dual role in coated vesicle formation, that it functions early as a regulatory GTPase, and this function is in its unassembled state, where it's targeted at early stages to clathrin-coated pits. And in this regulatory mode, it functions to regulate and monitor coated pit maturation and to control these slow stages of vesicle formation, requiring basal GTP binding and hydrolysis activity. It subsequently, after self-assembly, switches to its second mode of operation as a component of the fission apparatus. These late stages of dynamin function, the fission reaction is very rapid compared to the maturation process, requires self-assembly and dynamin's assembly-stimulated GTPase activity. So that's the take-home. Those of you who want to leave, I'll try to now provide evidence for that. So the evidence for dynamin's role as an early regulatory enzyme in GTP hydrolysis is actually quite extensive. First of all of the GTPase and assembly defective mutants that we and others have generated in dynamin over the past 10 or 15 years block early stages of vesicle formation, not late stages. Chemical inhibitor of dynamin, which is a non-competitive GTPase inhibitor identified by Tommy Kirkhausen, also blocks early stages of vesicle formation. As I told you, mutations in GED that impair assembly-stimulated GTPase activity accelerate these early stages in vesicle formation. A second class of mutations in GED that were identified by Drosophila genetics as suppressors of the Shibiri phenotype, so-called sushi mutants, suppressors of Shibiri. These mutations in GED rescue second site mutations in the GTPase domain, which shows that the GED activity negatively regulates dynamin function in vivo. We'll come back to these mutants a little bit later on in the talk. And finally, recent studies in our group, which I'll show you the data from, show that dynamin's basal GTPase activity controls the turnover of abortive coated pit intermediates. So this work that I'll take a moment to tell you about is work by Marcel Metland, a postdoc in my lab, in collaboration with Dina Locher, a postdoc with Gaudens Danuser, who was in at Scripps, and Gaudens has since moved off to Harvard heaven. This is total internal reflection fluorescence video microscopy of fluorescently labeled, EGFP-labeled clathrin light chain dynamics in a BSC1 monkey endothelial cell line. And what you're looking at here is with the turf, we can illuminate only the bottom 100 nanometers of the cell, and we don't illuminate any of the fluorophores deeper in the cytosol. So in this way, we can monitor events right at the cell surface. It's ideally suited for monitoring clathrin-mediated endocytosis and other events at the cell surface. Claire Waterman helped us set this up when she was at Scripps. She's now at NIH heaven. Anyway, what you're seeing here are coated pits that form and pinch off from the cell surface. And as you look at this movie, I was struck by the heterogeneity of events. There are ones that come and go very quickly. There are ones that hang along a little longer. There are very bright, intense objects, and there are dimmer objects. And so when I looked at this movie as a biochemist, I was a little disturbed, in a sense, by this heterogeneity, because as a biochemist, by analogy, we tend to take a mixture of components, a mixture of cells, a mixture of events at the plasma membrane, and put them together into one blender and end up with something akin to a tropical fruit smoothie. Well, in fact, so I, in a sense, as a biochemist, have been studying tropical fruit. And there may not even be such an entity as a tropical fruit. We study as biochemists the average of events. And in that averaging of all events, we may or may not lose sight of reality. And so our goal with this new assay was to capture and quantify the heterogeneity of events, and then to define the functional and molecular basis of this heterogeneity, and most importantly, to exploit it as a source of new mechanistic insight. So in order to do this, we needed a fully automated, unbiased way to track all of these events and to establish a complete inventory of trajectories by turf. And hence, we collaborated with Gaudens Danuser, Dina Locher, and Khalid Jacquemin from their lab, who had to deal with this problem of tracking coated pits. The problem is their density, their heterogeneity, and the low intensity of the objects, and also the fact that they might temporarily disappear because of unevenness in the cell or the turf image. And if you systematically lose sight of something and then gain it again, and if you don't fill in those gaps, then everything's going to look like it's short-lived. Right? And so the mathematics for this problem comes from astronomy, where astronomers will look up in the sky during the night and see all these stars of different brightness and heterogeneity and twinkling, and then have to go away for the day and come back the next night and figure out which star is which. Right? So there is mathematics to do this, and it needed to be modified, but Khalid Jacquemin and Gaudens and Dina did this in a way that allowed them to spatially and temporally globally optimize and track all of these images, including this all-important method of gap closing. When we detect and track and give trajectories for all these objects and cross the finish line, it's now important to apply statistics to interpret what this heterogeneity is telling us, and that was done with Bayesian information criteria. So what you see here is the analysis, the statistical analysis, I don't have the N here, of roughly 50,000 endocytic events taken from 24 different cells, 24 different movies, long and short movies. We're trying to capture all of this heterogeneity. And what we see is that there are three populations statistically of these dynamic behaviors of these coated pits. Two short-lived populations which we ascribe to as abortive events with lifetimes of 5 and 15 seconds, and about half of the coated pits have longer lifetimes that we were able to ascribe as productive events leading to coated vesicle formation. So we then took this new assay and we did a dynamin knockdown, not a complete knockdown, just about a 50% reduction. And I think this is important because we want to create hypomorphic effects. We don't want to completely kill endocytosis. We want to have a small effects and use this sensitive assay to pick it up so we can get more, rather than the potential of feedback, inhibition, and other indirect effects of strong dominant negative. So with a 50% knockdown of dynamin, as we expected, as I showed you earlier, dynamin is controlling the rate-limiting steps of vesicle formation, and the lifetime of these productive coated pits increases quite significantly. And this lifetime can be rescued, in fact, a little bit enhanced, by rescue with the RNAi-resistant dynamin wild-type protein. What was unexpected is, in addition to these effects on the productive population, we also found that knockdown of dynamin caused an increase in the lifetime of these abortive coated pits. So this suggests that dynamin was in part regulating the turnover of these early endocytic intermediates. So from this and a lot of other data that I don't have time to go into, published in these two papers in last year, we are proposing a conceptual framework in which to think about this GTPase and its regulation of endocytosis, and in which we're suggesting, that dynamin functions to monitor an endocytosis checkpoint. So the basal GTPase cycle of dynamin is controlling this checkpoint, and data I haven't had time to show you, and input into this basal GTPase activity and actually into dynamin's propensity to self-assemble are coat proteins, factors that recognize membrane curvature, factors that regulate cargo, that provide input into this checkpoint as a decision is made whether to abort early non-productive events or to continue on a maturation pathway towards formation of coated vesicles. So we are now busy trying to probe the mechanisms and the input into this endocytosis checkpoint as a framework for our thinking. And much more work needs to be done. But looking at this idea of dynamin as a checkpoint regulator, as a control of these abortive events, it reminded us of data that's out there for the SAR and ARF GTPases that control COP1 and COP2 vesicle formation. I think, in fact, Jennifer's work showed that the ARF was turning over more quickly than the coat proteins and at the Golgi, and in fact, it's now known that there are abortive events in COP1 and COP2 vesicle formation, and that the stability of a coat moving through maturation is dependent in part on the gain of curvature and the assembly of coats and the recruitment of cargo molecules. So we think that in fact these early functions of dynamin are quite parallel to the early functions of SAR and ARF in regulating vesicle formation at COP1, of COP1 and COP2 at the Golgi and ER. So now I want to talk to you about dynamin's later role in endocytosis as a component of the fission apparatus. And much of this work was done by two very talented postdocs in the lab, Rajesh Ramachandran, who left this week for the Physiology and Biophysics department at Case Western, and Thomas Pukadil, who'll be returning in the summer back to India to start his own lab. So there have been several models for dynamin as a mechanochemical enzyme mediating membrane fission. The first detailed model was put forward by Jenny Henshaw, having examined the structure of dynamin on lipid, assembled onto lipid tubules, or onto liposomes into these tubules, both in the absence of nucleotide and in the presence of a non-hydrolyzable analog of GTP, GMP-PCP. And Jenny noted that, that these tubules became constricted in the presence of GTP and suggested that dynamin functioned to squeeze the necks for membrane fission. Harvey McMahon also looked at different nucleotide-bound states of assembled dynamin, this time onto more rigid, preformed lipid nanotubules, and he found a structure much like Jenny's in the presence of this non-hydrolyzable analog of GTP, but when he assembled dynamin in the presence of GDP, he saw a more open helix, and he suggested that GTP hydrolysis caused a helical expansion that would pop off a vesicle from the membrane. In a more recent study, Pietro Di Camilli and Aurelien Roux in his lab pre-assembled dynamin, or allowed dynamin to pre-assemble on lipids that they were drawn out of a reservoir of lipids, and then they added a fiduciary gold bead onto these pre-assembled dynamin, added GTP to that, and they found that the bead spun around and twisted around the tube here. So they suggested that GTP hydrolysis drove a twisting action that would twist off these necks. However, all of these models are based on global conformational changes of pre-assembled dynamin. By and large, dynamin that's first pre-assembled either in the presence of non-hydrolyzable analogs of GTP or in the absence of nucleotides. Secondly, all of them conclude that dynamin alone was not sufficient to cause fission. So in Jenny's case and in Pietro's case, they suggested that external tension was required, that the two ends of the tubules had to be tethered onto something to get fission in the middle. And in the case of Harvey's model, he suggested that you needed to push against something like a coat proteins in order to get the popping to occur. There was no fission occurring in his isolated tubules. But the fact is that in the cell, dynamin is always in the presence of GTP, and as I told you earlier, with its 10 micromolar affinity for GTP, it's also pretty much always GTP bound. So how can you reconcile these models with that property of dynamin? So we wanted to look at the dynamic behavior, I think there's dynamic dynamin, but the dynamic behavior of dynamin on liposomes. And so then enter Rajesh Ramachandran, who set up real-time assays for dynamin activities. And he did this first by mutating all of the surface accessible cysteines on the molecule to serine, confirming that that this mutant of dynamin behaved exactly as the wild-type protein, both in vivo and in vitro, and then adding back a single cysteine mutant in different places in the molecule that he could then label with fluorescent probes. And the first fluorescent probe he used, labeling this isoleucine in a variable loop in the pH domain, was NBD. NBD is an environmentally sensitive probe. That is, it doesn't fluoresce in aqueous solution, but when inserted into a nonpolar environment like the lipid bilayer, you can see as you add dynamin, this fluorescently labeled dynamin to liposomes, you see this about 7-8-fold increase in fluorescence intensity. So what happens when you add nucleotides to this dynamin? In the presence of GMP-PCP, nothing happens. you basically stabilize dynamin on the membrane, and there's no further binding. That is, dynamin-membrane interactions are nucleotide independent, consistent with what Jenny had shown, for example. When you add either GDP or GTP, dynamin disassembles rapidly from the membrane, reaching a new equilibrium that involves reactions of assembly, hydrolysis, and disassembly. Introducing cysteine in another location in the molecule and labeling it with a different probe, in this case BODIPI, Rajesh developed an assay to measure dynamin-dynamin interactions on the membrane. So when dynamin assembles on the membrane, these BODIPI fluorophores come into close contact with each other, and you get auto-quenching. So once again, when you add liposomes, you see the quenching of BODIPI fluorescence due to dynamin-dynamin interactions. Now, in this case, when you add GMP-PCP, you see further quenching of the signal. Now, since there's no more binding of the molecule under these conditions, we interpret this as a conformational change that brings the fluorophores into closer proximity or angled to each other so that the quenching increases. And we think this is probably a fluorescent correlate to the constricted state that Jenny Hinshaw has published. When you add GTP, you see a transient move towards this conformational change, and then, as you expect, the disassembly of dynamin from the lipid bilayer. Comparing the kinetics of disassembly of the membrane, the kinetics of release from the membrane by the NBD dissociation assay, and the kinetics of disassembly of dynamin-dynamin interactions as measured by the BODIPI assay, you see about a 1 second lag here, in which dynamin remains on the membrane, but remains assembled, but releases the membrane. All right? So the question is, how do you reconcile this behavior to membrane fission, a mechanism for membrane fission? So enter Thomas Pucadil, who took a page out of a playbook of developing planar supported lipid bilayers. This has been around, method has been around for some time, in which you can take liposomes, absorb them to a glass surface. They fuse, and rupture to create a supported bilayer on these glass surfaces. This can be done with glass cover slips, but it also can be done with glass beads, silica beads, in which you can deposit a supported bilayer around the beads with a thin aqueous layer of about a nanometer, so that these lipids are free to diffuse within the plane of the bilayer in these systems. The problem is, when you make a tight, stable, supported lipid bilayer, there is no reservoir from which you might be able to remodel the membrane or pinch off vesicles. And so what Thomas developed is a new approach in which he deposits these liposomes on beads in the presence of a high salt buffer. And there's a paper that has just been accepted a couple of weeks ago in Biophysics Journal that reveals a mechanism of how we're getting this extra membrane deposited on these beads. But under these conditions, Thomas gets about a 3-fold more lipid on the bead than is required for a nice tight-fitting membrane. We think of this like pre-plastic surgery coated bead, little floppy there instead of the nice tight young coated bead. So you can see this reservoir here when we take these beads and allow them to sit on glass cover slips, and you can see here very fluid reservoir that spills out onto the cover slip. If you make beads in a conventional way, supported bilayers in a conventional way, there is no reservoir to spill out on these cover slips. So this now becomes a simple assay that we can use to visualize membrane remodeling, and the assay is quite simple. We take these super templates made from PCPS, PIP2, and spike in a little bit of rhodamine fluorescence. In the biophysics paper, we've used cholesterol and PE and a variety of lipid compositions. So this can be done with virtually any lipid composition. Anyway, you need now to use pegylated cover slips, because if you don't, the reservoir will spill out. So we use a pegylated cover slip. We just have a little drop where we put the beads on and we let the beads settle, and then we use a simple epifluorescence microscope with a 60x objective lens that allows us to visualize this field of beads. And then to this drop of beads, we add dynamin from a pipette and see what happens. So when we do this in the absence of nucleotide, you can see, hopefully, these lights might need to come down for some of these images, but you can see little hairy projections emerging from these beads. They get a little thicker. There's bundling, as you'll see in the next slide. And sometimes these things, as they get long, you can see this fraying and splaying apart, and eventually you can get a tether formed as these two interwrap and intertwine around each other. So this is in the absence of nucleotide, and you can see that the Bodipi dynamin is enriched. It's on the surface of the beads, but it's enriched relative to the lipid on these tubules. By EM, you can see that dynamin has assembled just as it does with purified liposomes. And you can also see that these tubules bundle and wrap around each other. In fact, dynamin was first identified as a microtubule bundling protein, and we think what it's doing here is bundling these lipid tubules as it forms them. Just in passing, we also think the twisting that was seen in those GTP experiments reflects the unraveling of these bundled lipid tubules, because these things unravel in the presence of GTP. All right, now in the presence of GTP in the real life situation, we have GTP in solution, then we add dynamin. You don't see these hairy projections, but what you see coming off the beads are small vesicles that appear. Over time, filling the media. There you go. So this assay, which allows us to visualize vesicle formation, also allows us a very simple biochemical assay for vesicle formation. We simply take the beads with a little bit of fluorescent lipid and incubate them with dynamin, with or without nucleotides and non-hydrolyzable analogs, and then sediment them for a couple of seconds in a microfuge tube and look for the released fluorescent lipid. And you can see that the release of fluorescent lipid into the supernatant is absolutely dependent on dynamin and GTP. Mutants of dynamin that are defective in GTP binding or defective in self-assembly are unable to release vesicles from these supertemplate beads. In addition, what Thomas has done is look at a series of dynamin mutants that differentially affect clathrin-mediated endocytosis in vivo, and you can see a very, very nice correlation between the ability of these mutants to mediate fission in this supertemplate assay and their ability to support clathrin-mediated endocytosis in vivo. So Thomas also developed a trick that allows us to visualize what's going on during these fission reactions. Because of the intensity of fluorescence of the lipid around the bead, we can't actually see what's going on at the surface of the bead as we make those small vesicles. But Thomas discovered if he adds a little bit of glycerol while he's adding dynamin, or in this case, just adding a little bit of glycerol, again, because of the fluidity of this membrane, the Schlieren force, the shear force, drives this huge, 1 micron, these are 5 micron beads, so this is like a 1 micron thick bolus of membrane reservoir just coming out and then flopping back into the beads. When you do this same thing in the presence of dynamin, but in the absence of nucleotide, that large vesicle, that large tubule is rapidly captured by assembling dynamin, and these bulbs at the end remain tethered. It's hard to see in these images, but there's a very thin tubule that's tethering, coated with dynamin, that's tethering this bulb to the supertemplate. In contrast, if you do the same thing now in the presence of GTP, you see these immediate constrictions. You can see this one's still tethered, that movie's still running. Immediate constrictions here, localized constrictions that then pinch off. This tells you that dynamin is a very, very efficient curvature generator. Within a second, it has assembled and generated, taken that 1 micron tubule and constricted it into a little tiny tether. That's an incredible amount of curvature generation activity. And it turns out that curvature generation activity is dependent on its ability to self-assemble, but that isn't sufficient. It's also dependent on insertion of a hydrophobic residue, a hydrophobic loop in the pH domain. So Rajesh identified a single amino acid residue, I533, which when mutated to an alanine, abrogates dynamin's ability to generate curvature. So did I run this movie yet? There you go. So here's a wild type. I want you to watch the fusion comes out, and this 1 point mutation, it can't capture these structures and generate tubules, whereas the wild type has rapidly captured and is now pinching off vesicles. So dynamin self-assembly and scaffold activity is necessary, but not sufficient for this very effective curvature generating activity of the protein. So again, using this same technique, and now diffusing in fluorescent dynamin. There's a bead here that you can't see. We're going to diffuse in fluorescent dynamin and watch what happens. You can see that the fluorescent dynamin, it binds to the beads, but it's enriched, highly enriched at that neck, where it stays in this localized constriction until it pinches off. There are puncta of dynamin around the bud and around the bead. These probably represent small vesicles being pinched off during the course of this reaction. Remember, in this case, the lipid is not labeled. So this is a time-lapse series from that same movie, and you can see that if we take it at just the contrast, we can see dynamin throughout absorbing on all membranes. But you can see that it's very highly enriched here at this neck, where it rapidly generates highly localized curvature. It hangs around there until the fission reaction. If you can notice, there seem to be fluctuations of intensity. It goes brighter and dimmer and brighter and dimmer, and Thomas quantified this on a number of these examples, and indeed he saw this flickering of intensity prior to the fission reaction. So we think that there are cycles of assembly and disassembly before a stochastic fission event. And you can get the sense of the stochasticity of this fission event in this assay, where he's drawn out tethers first and adds dynamin. You can see it breaks the tether, same amount of dynamin around, but it's breaking it 1, 2, 3. It's breaking it sequentially. It's not breaking the whole thing apart in one go. So these are quite stochastic fission events. So what's happening here to explain this behavior of dynamin in fission? And one thing is for sure, dynamin can mediate fission, but it's not very good at it on its own. And so, of course, we're busy trying to add other factors to make this reaction more efficient. But some insights into the mechanism of this stochastic dynamin-mediated fission came from a collaboration with Josh Zimmerberg here at NIH and Vadim Frolov. Vadim and Josh had developed independently an assay to look at dynamin assembly and dynamin membrane interactions, in which they draw a lipid tubule out of a reservoir, black lipid membrane reservoir, using a pipette, such that they can measure the conductance through this pipette, the capacitance through this pipette, which is a direct measure of the diameter or the internal diameter, internal radius of the tubule. So they can draw out this thing, measure the radius of the tubule, and add dynamin in solution on the outside. What they find is when they draw out these tubules and they stabilize at a certain diameter with a certain conductance, and when you add dynamin to this outer bath reservoir, you can see that the conductance drops, the tubule is squeezed in a dynamin-dependent manner. This is in the absence of GTP. In the presence of GTP, there's only a little bit of squeezing. We interpret this as small assemblies that won't give you such a dramatic squeezing of capacitance along the length of the tube. And then, again in a stochastic manner, this case 20 seconds later, sometimes 70 seconds later, sometimes 10 seconds later, a fission event, an abrupt fission event that they were able to show was non-leaky. When they shorten the tubules to give them a little bit more resolution of what's going on during these lags before fission, again they saw this cycles of squeezing and release before an abrupt fission reaction. So this coupled to some very elegant mathematical modeling and theoretical considerations of lipid and forces led Vadim and Josh to propose the following model for dynamin-mediated, or we think of it as dynamin-catalyzed, actually, membrane fission, because a catalyst just lowers the energy barrier for fission, and that's what we think of dynamin in doing. So in the presence of GTP, dynamin forms these short localized constrictions through that curvature generating activity. Upon GTP hydrolysis, as Rajesh showed, dynamin releases the membrane before it itself disassembles. That released membrane has two choices. It can relax, expanding outward, causing a failed fission event and having to recycle again in a stochastic process, or it can collapse inward, leading to a hemifission intermediate and fission. As we showed and Vadim showed, if you pre-assemble dynamin into a long structure in the absence of nucleotide and then add GTP, this will not mediate fission. The pre-assembled dynamin must first disassemble and start over again before it can get to the short prerequisite of these short helices to mediate fission. So this again is kind of reminiscent of SAR and ARF. SAR and ARF are also membrane active GTPases. They have hydrophobic peptides that are displayed on in a GTP-dependent manner from these small GTPases. And unlike dynamin, they only bind membranes in the presence of GTP because of this conformational change. But if you incubate ARF and SAR with lipid tubules in the presence of non-hydrolyzable analogs, they tubulate them just like dynamin. So we think that this localized assembly of these GTPases might be a general mechanism for fusion. The GTPase, the localization of SAR and ARF to the neck is done by GEFs that are restricted to the donor membrane, and gaps like the coat proteins or this curvature-sensitive ARF gap that cause disassembly in the bud. So in this way, these GTPases, membrane-active GTPases, can also be restricted to the neck. Dynamin is sort of a self-contained GTPase that restricts its own assembly at the neck, and hence a minimum fission apparatus. All right, so there's this enigmatic role of dynamin's GTPase in membrane fission. I've told you that GTPase activity is required for membrane fission, yet it can negatively regulate dynamin function in vivo, and that GTP hydrolysis drives dynamin disassembly and membrane dissociation. So how is this really working? Well, we don't know, but Francis Crick said, to understand function, you must study structure. And so Jenny Henshaw has done this extensively with dynamin, doing cryo-EM of these decorated tubules, and she's looked at the assembled state of dynamin on these tubules, identifying the GTPase head on the outer surface of these dynamin-coated tubules, a stalk made of the middle and JED domain and the pH domain interacting with the bilayer. She's also looked at the dynamin in the absence of nucleotide, in the presence of nucleotide, and compared these structures and seen significant conformational changes in these stalk regions that account in part for the constriction that she's seen. There have also been structures of the GTPase domain. John Kuhl and Dietmar Manstein solved the structure of the rat dynamin GTPase domain shown here, and the dictyostelium dyne GTPase domain. It has the conventional GTP binding elements that all GTPases have, and we, based on these structures, busily mutated just about everything we could think of in the active site over a 10-year 14-year period, and none of these mutants gave us the kinds of phenotypes we were hoping for, which are mutants that are specifically defective in GTP hydrolysis and not GTP binding. We were not able, through these studies, to identify the catalytic mechanism of dynamin's GTPase activity. The same group, however, noticed that if you turn this molecule around and look at the back end, the C-terminus of the GTPase domain and the N-terminus of the GTPase domain form a hydrophobic platform in their crystal structure onto which was docked alpha helix from the fusion protein that they'd used for expression of this construct. So, I told you about these sushi mutants. It turns out they map to the very C-terminus of GED to a very conserved hydrophobic amphipathic helix with a very conserved hydrophobic face. And so we reasoned maybe that this N and C-terminal GTPase domain might provide a site for docking of this GED C-terminal helix. And then entered Josh Chappie, who was a graduate student in my lab starting in January of 2005, who tried to take on this project of solving this structure. He was unsuccessful, got married, moved to NIH, and completed the last year of his PhD here at NIH with Fred Dyda, and became a much happier camper. So what Josh did was to design a minimum GTPase GED fusion protein in which he took the GTPase domain, a small linker, and the last 26 amino acids, 25, 26 amino acids of GED. We call this GG. Unlike dynamin GTPase alone, this GG construct is fully soluble in, after cleavage of the MBP, and it has the same basal GTPase activity as full-length wild-type dynamin-1. If you mutate the GTP binding residues, you lose activity. Just before Josh left my lab to come to Fred Dyda's lab, he discovered that GG dimerizes in a GDP aluminum fluoride dependent manner. That is, in the presence of non-hydrolyzable analogs of GTP or in the absence of nucleotides, GG elutes on this gel filtration column as a monomer, but if you add transition state mimetics, GDP aluminum fluoride or GTP aluminum fluoride, now the protein shifts into a very stable transition state dimer. So it was this construct that Josh brought with him, and together with Fred Dyda, they were able to crystallize and solve the structure of this GG dimer. And I'm just told that this paper came out today online in Nature, so you read it. I don't have a movie, but it looks like this. It's a GTPase dimer, bundle signaling ion, so I'm going to take you through this. I'm going to tell you that this structure reveals the mechanisms of basal and assembly-stimulated GTPase activity, what it tells you about this bundle signaling element, which we've come to call the GED, N, and C terminal GTPase domains, and the implications for dynamin function in the last 2 minutes. So, basal GTPase activity, we were looking for that catalytic arginine. We mutated every arginine anywhere near the active site, and even a bunch of ones that weren't even close, with no luck. But it turns out in the crystal structure there's a bound salt exactly in the position of where you would need this charge stabilizing cation, and in fact with these two catalytic waters, so this suggested that the sodium was acting to stabilize charge. And indeed, if you look at the basal GTPase activity, you find that it is salt sensitive. So potassium, the physiological salt, is a little better than sodium, but with these larger salts, rubidium or cesium, you lose basal GTPase activity. So it's a bound sodium that's held in place primarily by the nucleotide and by backbone carbonyls here, explaining why we could not mutagenize to create a defect in binding and not GTP hydrolysis. The mechanism of assembly-stimulated GTPase activity is happening because of the insertion in trans from the other to sister of this dimer, of this D180 from the dimer partner into the active site. And this, you can see this series of hydrogen bonding interactions that reorient and stabilize the catalytic machinery. That's why we get about 100-fold stimulation. You see switch 1 moving into place, the P loop, switch 2, positioning everything for optimal catalysis. Josh identified 3 residues that look key to this mechanism for assembly-stimulated GTPase activity, and they are absolutely conserved amongst the dynamin family of proteins, but not in more distantly related human guanosine-binding protein or the bacteria dynamin-like proteins. So we mutated those, and aside from some aberrant thing we really don't understand on basal GTPase activity, these mutants do not inhibit basal GTPase activity, but especially the Q40E and the D180N, they virtually abrogate assembly-stimulated GTPase activity. So this gives us a mechanism specifically for assembly-stimulated GTPase activity, and we're now in the process of studying the phenotype of these mutants in cells. So I want to tell you about the bundle signaling element here, which exactly as Josh had proposed in his paper, is a 3-helical bundle of the N and C terminus of the GTPase domain and the C terminus of JED held together through these hydrophobic interactions. What's striking is if you look at the orientation of this bundle signaling element in the GG dimer, you can see they're pointing in 90-degree opposite directions. Well, the implications of this are profound. First of all, it tells us that the GG dimer must occur between 2 tetramers, because otherwise they have to be pointing in the same direction. So the GG dimer minimum is a dynamin octamer. In addition, if you look at Jenny's reconstruction in GMP PCP, remember an analog that does not support dimerization in Josh's assay. Once again, these neighboring subunits of dynamin are all oriented towards the membrane, and so it's impossible for these proteins to take on this transition state dimer conformation and for both of them to be down on the membrane. So this may explain that GTP-induced disassembly that we've been seeing all along. So how does this relate to membrane fission, these conformational changes? So in my very low-resolution models, you can see that dynamin here is the tetramer, that this is the unit tetramer, and upon GTP binding in the transition state, there must be a conformational change that involves a rotation and a collapse inward of these assembled structures. If we look at the on-side view, we think that GG dimerization is occurring through longitudinal rather than lateral interactions, that is, across rungs of the spiral, so that in this transition state, again, you have a destabilization of interactions with the membrane and a release upon GTP hydrolysis. So to be even more speculative, we think it's possible, given that dynamin has this insertion of a hydrophobic loop of its pH domain into the bilayer, that during this transition state, these molecules that form a very short collar around the neck might tilt in towards each other and cause tilting of the corresponding underlying lipid, buckling of the membrane to bring them into close apposition, and perhaps this critical diameter, internal radius, which is about the radius of a monolayer, that's needed then for spontaneous fission to occur. So this is a model, it's speculation, and now my lab is busy trying to use and develop fluorescence-based assays to use these kind of mutants in our fission assays, and always go back into cells to see what that means as we try to understand this mechanism of membrane fission. So I'll stop there. All that's left to do is acknowledge the people who have done this work. I'd mention them as I went: Josh Chappie, Marcel, Thomas Pukadale, Rajesh Ramachandran. Others are working on other aspects of clathrin-mediated endocytosis and different isoform differences with dynamin, lots of other interesting things going on that we couldn't put into here. I have excellent technical support with Marilyn Leonard, Charmy, and Vasil Lukianchuk that have really keep the lab operating very, very smoothly, and collaborations with Dina Luker and Gaudenz Danuser, which continue as we try to unravel the heterogeneity of coated vesicle formation. Josh and Fred, I hope, will continue talking to me now that Josh is happy again, and same with Vadim and Josh Zimmerberg. We're continuing work. So thank you very much.
Getting back to the assay where you're just monitoring by turf the budding events, have you ever looked at the impact of particular types of cargo, overexpressing different cargoes on that process?
Absolutely. So Jennifer's asking about the effect of cargo on the dynamics of coated pits. And in fact, we've looked at two constitutively internalized cargo. We've overexpressed the transferrin receptor and the LDL receptor. And interestingly, we see two different phenotypes with those two different constitutively internalized cargo. They use different adapter proteins, so we think that the exact complement and playing with the complement of cargo and adapters is going to impact on the dynamics of coated vesicle formation. And so we're studying that. We're very interested in that. And that's, I think, especially relevant when you start thinking about signaling receptors and how they might be talking to. I used to think coated vesicles were buses, and the bus kept going, you either had a ticket or you didn't. But I think they might be more like taxi cabs or maybe airport shuttles, whether it's the Washington Flyers, where they're a little bit more customized in where they go and how fast they go. And I think there's a whole iceberg of information and buried underneath the heterogeneity that we have so far been ignoring.
So you mentioned about the model on how the fission occurs. How about the early stage on membrane invaginate? What dynamin play and how you would imagine it works on that early stage?
Right. We don't think that dynamin is involved in the early stages. We've looked at our knockout. We've made dynamin knockout cells, and we accumulate coated vesicles that are already quite curved. So we think those early curvature generation processes are largely the coat and potentially some of these bar domain containing proteins. But we do know that the bar domain containing proteins provide input into dynamin as a checkpoint regulator, controlling both positively or negatively its self-assembly. So if you want to think of a GEF or, in terms of activating dynamin, these bar domain proteins can either say don't assemble yet, or go ahead and assemble. And so we think there's information there for the checkpoint. It's totally a hypothesis, but we're now trying to tease that out.
So how far can you go in rationalizing the results that were seen by various other people in light of the new information? In particular, the DeCamilli twist model is intriguing. Do you think the two strands, as these longitudinal dimers break up, can the strands walk around each other?
Is that with or without a beer? Without a beer, I will go this far. No, I think that if you look at the movies from Pietro's, that paper was spectacular, and I think that paper clearly showed that dynamin has mechanochemical activity, no doubt about it. But if you look, you can see first it's DIC. So first they have very, very thin tubes, and the ones they finally study are quite thick by DIC. So I think they have bundled. And we do see unfraying of our bundles when we add GTP. And what happens when we add GTP to a pre-assembled tube, the dynamin falls off, as we expect. And now, if they're tethered, the tube wants to shrink because it no longer has the extra surface area being given by dynamin. And so it's shrinking, but when it's tethered, it can't shrink, and so it ruptures in the middle. And that's what we see. We published these movies in Thomas's paper. So we think that that's what's going on. I don't think Pietro would agree with that, or Aurelian. And you know, we do see this transition state suggests there's some twisting going on. So I think, you know, even without beer, I would say that we still don't know, and there are many, many more studies to be done. And every time I make a prediction about this protein, I'm wrong, almost every time. So we'll see.
Okay, on that note, we can all adjoin to the library over here where there's a reception for Sandy with coffee and cookies and things. Maybe we can